So hello students, now let us take up a topic on cilium. This will be in locomotion. If there are many in this, we call it as cilia. Single one is called as cilium. There is another word that we often come across, which is flagellum. The internal structure of both cilium and flagellum in all eukaryotes is same. Cilium are usually smaller in size, lengthwise. Flagellum are longer. Cilium many will be there. Flagellum usually one or two only per cell or per organism. That is the only difference between cilium and flagellum. Internal structure in eukaryotic organisms is the same. And you see, we will get this in amoeba. We will get this in paramecium. We will get this in other ciliates such as Opalina, for example. Both these are protozoans. We also get this lining up bronchi in lungs. So even mammals have the same structure. So see how structure of cilium is the same, whether it is a human being or whether it is a protozoan. Just think about it. How by way of evolution, certain structures which have a particular use, that structure is highly conserved. So structure of cilium in protozoan is the same structure of cilium in human being, in say bronchi for example of lungs. It's also present in reproductive. They are the same. Similarly in sperm of human beings, for example, the internal structure of the tail, that is the same. It's just like a flagellum. Same structure. So what does this consist of? In paramecium, suppose we are examining it. So this is how generally we show the cilium. Remember that cilium are present all throughout the body, and on a piece of paper or on this board, it's not possible to show a 3D view. So remember that this is on all the sides of the body. So even in the front over here, cilia are present. Everywhere cilia are present. In paramecium, this part is called as oral groove. So there are cilia present everywhere. And although this is a protozoan, this will move very intelligently. It can move towards the stimulus. It can, if you keep some poisonous substance, it will try to move away from it. It will try to take food only through the oral groove. So it knows. We use the word know in a proper sense over here. It does not have a brain, but it still can avoid toxins and poisons. It knows to go towards food. It will avoid those stimuli which are harsh, and it knows to protect itself. So within that, how the cilia are able to function. So this part over here, for example, So this is the membrane of the cell, the plasma membrane. This envelops now that extension of the cilium, and inside the cilium, here we will be able to see these are microtubules, and they all originate from here. This structure is called as basal granule. This structure over here is called as basal granule, and these are all arrangement of different types of microtubules. This central shaft is also called as axoneme. Neme means thread because this is a thread-like structure, and axo because axis, so it's called as the axis. So if we take a section over here, a transverse section, it will appear like this. Now the microtubules have got arrangement of nine plus 2 arrangement. So 9 are peripheral and 2 are central. So these peripherals, they are arranged in pairs. We call them as doublets. So each of those 9 will have one like this, okay, and one like this. So each one of this is called subfiber A. This is called subfiber B. This is complete and this is incomplete. And here they will contain arms. So microtubules are 9 peripheral doublets and 2 central, which are separate. Each doublet has complete subfiber and incomplete subfiber. This is subfiber A, this is subfiber B, made of protein called tubulin. Each doublet is connected to central microtubule by spoke. So these are all the spokes. Each doublet has arms made of protein called dynein. These arms, which are made up of dynein, they have got ATP motors. Okay, so these dynein arms can move up and down because they can bind and unbind. They can move up and down. This causes cilia to bend to one side, and this is how it causes movement. Now if this is, say, a membrane and this is the cilium, let's say this is position 1 and it moves in this direction. This is 2, this is 3, this is 4. So this is 1 to 4. This is called as effector stroke. So the effector stroke, and when it now comes back, (5 to 8) is called as recovery stroke. So there is effector stroke and recovery stroke. Effector stroke is powerful. At that time the cilium is flat and straight, but when it is recovering it can become limp. So these two strokes are there when the cilium is beating, and all the cilia which are there, they may move in phase. All cilia, they may move in phase, but sometimes they are slightly out of phase. This is what happens often. So we get asynchronous movement of the cilium. We get asynchronous movement of cilium. Remember that suppose this is paramecium, the cilium on which we have shown over here left hand side and cilium on right hand side, they may move not in the same manner. They have to move in such a manner that the animal either moves forward or backward. So even though we say that paramecium or protozoan is simple organism, yet there is a level of sophistication in how it tries to protect itself and survive. So all these basal granules, they are interconnected by a network which we call as infraciliature, and they all get connected to a bilobed structure that we sometimes refer to as brain, and which is near the oral groove. But this is only speculation. There might be a structure like this which is similar to a brain. That word brain is just used because it happens to control the movement of cilia in paramecium. As I said, cilia are present even in human beings. So all organisms will have cilia, but the internal structure of cilium of all eukaryotes is the same. So with this, we come to the end of this topic on cilia. Thank you.